This week we were learning about the digestive system, which is basically made up of three basic phases. The cephalic phase, which is the sight, smell, taste, or thought of food that stimulates the parasympathetic nervous system to begin gastric acid secretion. This is followed by the gastric phase, which is once food is actually digested and it moves from the esophagus to the stomach, which the gastric phase is either triggered by the expansion of the stomach or once the food is in the stomach and starts binding to the receptors. During the gastric phase, food is further broken down by both the secretion of stomach acid and by digestive enzymes. And then once the enzymes, the acid, and the food is all mixed together and digested, it is deemed chyme, which the passing of chyme from the stomach through the pyloric sphincter into the intestine begins the intestinal phase, which is the beginning of nutrient absorption and then eventually the passing of food in the small intestine. Stomach storage is often a very overlooked aspect of digestion. It's really important because once the chyme reaches the small intestine, it needs time for nutrient absorption. And if what we ate directly passed from the esophagus, through the stomach, into chyme, and into the small intestine, continuously, we wouldn't be able to absorb all the nutrients and we would end up passing most of what we ingest, resulting in diarrhea. Therefore, it's really important to have this storage in the upper area of our stomach so that we can slowly digest our food and absorb the majority of the nutrients in our food and pass it healthily, and therefore we eat food, efficiently. So some of the important parts of the gastric system come from the basic anatomy of the stomach lining. So the lumen of the stomach is lined with epithelial cells that produce mucus, and this is used to protect the stomach from the low pH of the gastric acid. And between these epithelial cells are gastric pits, which lead to gastric glands. And these gastric glands have multiple cell types that all secrete different hormones, enzymes, gastric acid, and paracrine molecules that are all important in the regulation of the digestive system, and in this case, the gastric phase. So it's really important to know the function of a lot of these molecules. So some of the most important ones is the mucus, which is secreted by the surface cells. This mucus, again, serves as a barrier for the acid. The epithelial cells produced bicarbonate, which we actually used as a buffer in one of our first labs in class. And as we saw in the lab, it serves to lower the pH of acid. And this buffer is in the mucus along the lining of the cells, and it lowers the pH from 2 all the way, or higher the pH from 2 all the way to 7, making around the cells less acidic. That way we don't cause damage to those cells. Another important secretion is gastric acid. This actually leads to killing the bacteria and any microorganisms we may have ingested in the food that could potentially cause harm to us. And it also activates pepsin and turns it from pepsinogen to pepsin. This is secreted by the parietal cells. Also secreted by the parietal cells is the tryptophan factor. This combines with B12 and is a really important constituent in vitamin absorption once we're in the intestinal phase. Histamine is a really important secretion that stimulates the gastric acid secretion. So this is one of the paracrine molecules that leads to further gastric acid production. And pepsin and gastric lipase are both secreted by the chief cells. These are enzymes. Pepsin works to break down proteins, and the lipase works to break down triglycerides. Somatocin is a paracrine molecule secreted by the D cells, and this actually begins a negative feedback loop that ends the gastric acid production, which is really important because we don't want to ever have an excess of gastric acid. This is what causes indigestion or ulcers, and it's really important to have this somatocin that creates this negative feedback loop to end the gastric acid production. What actually begins this whole process is gastrin. Gastrin is secreted by the G cells, and it's a hormone that is released into the bloodstream once there's a presence of amino acid or peptides in the stomach. Once the G cells release this hormone into the bloodstream, it directly stimulates the parietal cells to release the gastric acid, and it also causes histamine to be released, which triggers more gastric acid secretion. So you may be thinking, well, if food is needed and amino acids and peptides are needed in order to trigger the secretion of gastric acid, how is gastric acid secreted in the cephalic phase? This is actually because gastrin can also partially be released by neural reflexes called gastrin-releasing peptides. And this is when your parasympathetic nervous system stimulates the release of gastric acid due to your thought, taste, smell, or sight of food. So once the parietal cells are stimulated by gastrin, it secretes gastric acid. Gastric acid has many functions. One of the functions is it causes the release and activation of pepsin, which is what breaks down proteins. It triggers somatocin, which eventually is the negative feedback loop that ends the gastric acid production. It causes the gastric acid, hydrochloric acid, which denatures proteins, which breaks them out of their tertiary structure and makes them more easily digestible. It also helps to kill bacteria and other digestive microorganisms, which can be very harmful to us. And it also inactivates salivary amylase, which stops the carbohydrate digestion that begins in the mouth. So this is a list of a few of the constituents that are released during the gastric phase. The pepsin is what is initially released as pepsinogen by the chief cells, but once it's released and comes in contact with the acid, it turns into pepsin and is activated and is able to break down proteins. Lipase is the enzyme that initiates a breakdown of triglycerides. Histamine is what stimulates parietal cells for acid secretion. The intrinsic factor combines with B12 to aid in vitamin absorption, and somatocin is secreted by the D cells to start negative feedback loop, which in turn ends the gastric acid secretion. So some interesting research about the digestive system is that the excess production of acid is actually not the most common cause of peptic ulcers, which is a common misconception. Actually, non-steroidal anti-inflammatory drugs such as aspirin are the most common cause. So if the bicarbonate buffer of the epithelial cells is not able to efficiently buffer the low pH of the gastric acid, this acid can destroy some of the epithelial cells, causing indigestion and ulcers and severe discomfort to a patient. Initially, antacids were the main treatment for this excess acid secretion, but upon the discovery of the mechanism for acid secretion through the parietal cells, it became obvious that two better therapies were possible hydrogen receptor antagonists and proton pump inhibitors. And these are the main targets of treatments used today. In parietal cells, once parietal cells are activated, the process begins by H+ from water being pumped into the lumen by the ATPase in exchange for potassium entering the cell. Chlorine ions then flow into the cell through chloride channels, and this results in the hydrochloric acid being secreted by the cells. So what the hydrogen receptor antagonists do is they work against allowing the hydrogen to come into the cells, and the proton pump inhibitors also inhibit the hydrogen being pumped into the cell, which would then in turn inhibit the hydrochloric acid secretion.